Thank you very much, David. And do I do I put the slides up or do you do that? Because I can put them up.
Yes, feel free to
Okay, I
feel
will
more
do that.
comfortable to control your slides.
All right. I should be able to do that. I just hope uh, I don't waste much time. How does that look?
Yes, we see. Great.
Excellent, excellent. All right. So I think I'm all set. And uh, implementation science has so many different examples that one could use from so many different fields. But since we share an interest in chronic infections like HIV, acute infections that have chronic manifestations like SARS-CoV-2 causing COVID-19, Rick and I thought that it would be reasonable to give you an example of how one assesses community effectiveness of vaccines and giving you a real world example from the past few years. So I'll talk a brief introduction to translational research, try to put the context of implementation science in the context of translational science, give you this virology translational research example from a vaccine effectiveness study in the Dominican Republic. I want to tell you a few words about a large network, and we're very interested in seeing if Georgia would be interested in joining this network, the Global Virus Network, and a little bit about the need for translation and implementation research in pandemic preparedness. So it may be that you already saw this cartoon because I think I may have gotten it from Luke Davis. The latest research shows that we really should do something with all this research. And then here's one for Rick and myself. Shoot, we'd better hurry up and figure this out. This is two aging researchers. So why do we need implementation in translational science? Because we want science to actually make a difference in the lives of people. And two papers, Lawrence Green's paper from 2009 and Zoe Morris's paper from 2011, highlighted how long it takes for discovery to translate into actual practice. And I won't read this slide, but just to give you a sense of all the different steps that cause an unacceptably long time between research initiation and actual deployment of discovery in practice. I was early in my career at the National Institutes of Health working in the Division of AIDS, and I saw the sense of urgency and the anger in the community of persons infected with HIV, demanding that the NIH and the Food and Drug Administration accelerate their research timetable and accelerate their review timetable to make life-saving antiretrovirals and other drugs available to people who are dying literally of HIV. And I'll never forget my experience at that time because at that time I became a complete adherent to implementation science, even though it had different names back then, because I actually agreed completely with the activists. Now, here are some historical examples of both slow and fast implementation. Way back in the late 18th and early 19th centuries, you had Lady Montagu, the great advocate for variolation, actually using smallpox from variola minor to immunize against variola major. Problem with that is you didn't always guess right, and sometimes you actually gave people variola major. But then Edward Jenner's observations that the cowpox actually was protective against smallpox, and it took more than 50 years for that to enter standard practice. And this was in the era where microbiology was in its infancy. Penicillin also took an unacceptably long time from the discoveries of Fleming then chain, then flurry. And it really, when you go back into the history, it was really an outrage that it took more than 15 years. Insulin, on the other hand, this instantaneously life-saving drug derived from human and animal sources, the Canadians, Banting and Best, this was translated into practice in 6 months. So if you have something like insulin or you have people dying of HIV, you may be able to accelerate things. Now, this I borrowed from a site that I found at Harvard University, and I edited it a little bit. So you have pre-intervention. Is there a relationship? Efficacy studies, can programs work? Effectiveness studies, does the program work? And then implementation studies that we're studying in this GIFT activity. And it's very helpful to consider implementation from the outset so that the pragmatic trials or collecting pilot implementation data are done thinking about the ultimate application. And translational research we think of as bringing basic science to the clinic, bringing the clinic to the community, and then scaling up in the community to have a nationwide and global impact. And you can think of these transitional research efforts as translational science 1, 2, and 3. And sometimes you just map it to basic science, clinical science, public health science. So I saw this at Virginia Commonwealth University and borrowed it. Translation between basic science in medicine, engineering, economics, and the life sciences, life, physical, computer, social sciences, into improved or novel treatments and interventions, including prevention. Translate that into community application, penetration, cooperation. And if you made another step to the right, you would talk about scale up and implementation science. So translational science and microbiology, we need to take laboratory and animal research to clinical use. We need to bring clinical science that is promising to community application, and then ultimately the implementation science step, clinic and community to scale for impact at a clinical and public health level. So bringing these initiatives to scale is the key concept. And when is basic research ready for translation? Well, we're always struggling with cell culture and appropriate cell culture use. Which one is most applicable to human circumstances? Appropriate animal models. If they don't exist, then it's much more difficult to make that leap into human studies. Efficacy and safety must be demonstrated in animal models and in cell culture systems. And sadly, there are no clear established guidelines for when basic science is ready for translation. So we tend to reinvent the wheel every time. Now, what factors are needed to design basic experimental work to plan for later translation? And cell cultures and animal models, as I mentioned, assessing advantages of drugs, immunotherapeutics, vaccines, and other biologics of interest. You don't want to start on a path where you're less likely to have something that is competitive with current interventions. And you need to document the mechanism of action as best you can. That will help with assessment of potential side effects and interactions. And finally, collecting data with Food and Drug Administration or the European Medication Association, various organizations that guide us. And we in the United States have clinical translational science awards that help us liaison to these FDA caliber trials. And you want to get into phase 1 human clinical trials and ultimately phase 2, phase 3, and even post-marketing surveillance. Now, why is translational research in virology often elusive? Well, I mentioned cell cultures and animal models. You need to try to find the right model, ask the right questions. There are no NIH criteria for translational work in grants, so we have to promote the best lab and practice standards as best we can. There is a substantial gap in the physician-scientist workforce. There are just too many attractive alternatives for doctors in clinical medicine, in the farm industry, in administration. So in the US, we're looking at centers. CIRA is a good example, and I think we've described to you what CIRA is. CIRA centers to create collaborative dynamics and incentives to make it easier for people like myself or Rick you know, our other colleagues who are MD based researchers. And then the K23 awards that you'll probably be talking about in this session that are now available to international scholars, so-called NIH Career Development Awards, but through the NIH are available to international scholars, which can really improve the ability of folks to start a physician scientist career. And this failure of MDs and MD scientists to engage with each other, that's another element where we nurture interactive meetings, seminars, projects between one another, and GIFT is an example, and CIRA is an example. Now, how do we design clinical trials to inform basic science? So can you take clinical studies or public health studies and bring specimens back into the lab and bring questions back into the lab so you create feedback loops? So T2 and T3 science starts feeding back into T1 science. Now, basic scientists can join clinical researchers in the design, follow-up, and interpretation of clinical trials. We can have influence on the quantity, type, and cost of sample collection, try to collect samples that are going to be useful for laboratory scientists after the trial is finished or during the trial. And if we can manage subsidies from research grants for satellite research projects, maybe we can get indirect cost returns that are provided by some agencies. Maybe we have enlightened trial sponsors that are willing to support these satellite research projects. And we also have to plan what we can learn from negative trials. So if you're doing a vaccine study, you need to start thinking, what if the vaccine doesn't work? What kind of lab studies can I be doing to find out why it didn't work? Because that can be hugely beneficial as we go forward in vaccine studies. And the same is true for therapeutics, immunotherapeutics, even diagnostics that don't work well. Why didn't they work well? Can you design the laboratory studies to answer that question? Obviously, if it works, great. But I'm keen on learning from negative studies. Now, there are many new platforms of big data, artificial intelligence, machine learning. Many of them are mediated through integrative structural biology. And CTSAs, Clinical Translational Science Awards in the US, provide a platform where scientists can examine clinical trials, consider important ancillary studies when these CTSAs are doing what they're supposed to do. And we can have conscientious efforts for universities to nurture the engagement of MDs and PhDs. Now, open access to data, especially between large networks that have invested sometimes hundreds of thousands, millions of dollars in studies, so that you can easily find and understand a trial. You can easily find and access specimens that were collected during the trial and the data from the trial, including beyond one's own institution. And I wish we were doing better with this, but I think we're doing better now than we used to. So that's just a few words about translational research in the context of implementation science. Now I'd like to dive into the example for the day, virology translational research from a vaccine effectiveness study in the Dominican Republic. So back in 2021, we initiated a study with colleagues in the Dominican Republic to determine the effectiveness of the vaccine that they used in real world field conditions. And they were using the Chinese Sinovac Coronavac inactivated vaccine. That was one of the very first that was actually developed, sort of a classic Salk type vaccine where you simply inactivate the organism. And we knew already that it wasn't as immunologically potent as the mRNA vaccines were. However, about half of all the doses that had been given in the world up to that point were vaccines. So there were people who argued, well, we should have waited for the better vaccine. Why did we use this less immunogenic vaccine? But others said we should use what was available to us, what was affordable to us, and that's what the Dominicans had said. But all of us would agree to answer, were these vaccines worthwhile or were these a waste of time? What is the evidence that it actually made a difference in real world settings? So the methods were what we call a test negative case control study. I'll tell you what that is in a minute, to assess the real world effectiveness and using this coronavac on 3 outcomes: symptomatic infection, hospitalization, and death. And so participants suspected of having COVID-19 were recruited from 10 hospitals in 5 provinces. And that meant you had some respiratory symptoms that met the Dominican national criteria for a suspected COVID case. But the people with these symptoms could very well have a bad cold, could very well have influenza, could very well have RSV, rhinovirus, all sorts of respiratory viruses. So when we tested them with PCR, the positive PCRs were the cases and the others were the controls. So that's what we mean by a test negative case control study. They're all symptomatic, but the cases are the COVID positives and the controls are everybody else. So then we assess their vaccination status. And some were fully vaccinated with 2 vaccines and some were only partially vaccinated with 1. And vaccine effectiveness was estimated with multivariable adjustment for individual demographic and clinical characteristics. This is what the Dominican Republic looks like. Here is the Caribbean. Here's Florida in the US, Mexico over here, Central America, Venezuela over here, the Lesser Antilles, the Greater Antilles. Here's Jamaica, here's Puerto Rico, here's Haiti on the same island of Hispaniola as Dominican Republic, and here's Cuba. So you know where Dominican Republic is. And these were the parts of the country where we were working, where the 10 hospitals were located. So we had some diversity in, of location, which we thought was important. Now, this was the effectiveness of coronavac against symptomatic COVID-19 illness. Now, somebody's probably already told you this, but we like to use the word efficacy if we have an idealized randomized clinical trial, because then we are pretty sure that the 2 groups are comparable, the vaccine recipients and the placebo recipients, or vaccine A recipients versus vaccine B recipients. But we use the term effectiveness if we're looking at real world conditions. And Luke Davis can go into a lot more detail on this. So the point is that we found in adjusted odds ratios that, not surprisingly, the elderly were more likely to have had symptomatic COVID-19 illness, but the vaccinated folks versus the unvaccinated had protection, which was really quite impressive. 0.5 odds ratio for partially and a 0.7 odds ratio for fully vaccinated individuals. And this translated to a vaccine effectiveness of somewhere between 30 and 50%. Now, we're not so impressed with symptomatic disease. That's hard to measure. And some of the non-COVID cases might have some impressive symptoms as well. And at the end of the day, it just is not as important an outcome as hospitalization. Are we actually protecting people from hospitalization? And for hospitalization, we had age as a very, very powerful predictor with an odds ratio of 4, and obesity and comorbidities also contributing. But the vaccine was impressively effective at 70 to 74% effectiveness for partially or fully vaccinated persons. And the 95% confidence interval for fully vaccinated persons was fairly narrow at 62 to 83%. So I think we answered the question. Yes, it was worth vaccinating people using the inactivated product, even though the immunologists were not impressed with it, and it did not seem to have anywhere near the immunogenicity in the laboratory that the mRNA did, but it was hugely beneficial at the time that the ancestral strain and the Delta strain were circulating. So that was helpful, and WHO was pleased to have these results because they needed to know whether the billions of doses being given had done any good or not. Now, the Dominican Republic findings supported quite a number of other studies, including Hada et al. In the New England Journal from Chile. And they didn't use a test negative case control study. They actually used a true cohort in the country. And they were able to assess this and found very, very similar effectiveness data than we did. And in Brazil, in Indonesia, and in Turkey, similar studies to ours, test negative case control studies, found similar results. So it clearly indicates that lives were saved and hospitalizations avoided with the early deployment of this vaccine. I didn't show you the death data. They showed strong protective effectiveness, but because we didn't have so many deaths, it was not statistically significant, but the trend was in the same direction. So widespread use of vaccination is obviously a major tool for reducing serious COVID-19 disease and death. And the countries that decided to deploy the early available affordable vaccine actually made a pretty good decision in our judgment. So there's an example of implementation science that can really help guide us in future pandemics. If we have a vaccine that is not as immunogenic, but it's able to be deployed affordably and quickly, it may very well be worth doing. So we thought this was a helpful study, and that's published last year. Now, an opportunity arose while we were doing our study, and that is the Dominican Republic chose to boost with heterologous vaccine. They purchased several $1 million worth of the Pfizer mRNA product. And Eddie Perez Sten, our colleague in Dominican Republic, came to me one day and said, is there anything we can do with this? And I said, something, I have a friend, Akiko Iwasaki, at the Yale School of Medicine, and she's doing work in exactly this particular area. She's extremely interested in variants of concern, and she's working closely with a number of clinicians, Albert Coe and Saad Omer and others, looking at transmissibility, severity, vaccine effectiveness, efficacy. And I can call her up because if we can get a specimen before they're vaccinated and then a couple of times after they're vaccinated, we can compare the boosting effect of the mRNA against other boosters that have been used here in Dominican Republic. So we obviously are interested in the spike protein and antibodies against the spike protein. We have the ACE2 binding sites. There's the N-terminal domain, the fusion protein, and transmembrane anchor. And this is the interesting part about the SARS-CoV-2, is it has these unfolding and folding functions and also locked functions. And critical work from McClellan et al. From Dartmouth early in the pandemic taught us how to manipulate the mRNA to make the antibodies effective at responding to the SARS-CoV-2 when its unlocked mechanism made it susceptible to antibodies. So the emergence of Omicron and the question of whether previous vaccines were going to be at all helpful kept emerging. You had in the UK, in South Africa, in Brazil, in India, all through latter part of 2020. And the Omicron was first discovered in South Africa and Botswana, following up these early variants of concern. So our first sort of reverse translational study, where we took an advanced time of deployment, namely post-vaccine effectiveness evaluation, and we ratcheted back to T1 research, was we asked the question, what's the effect of mRNA boosting in persons who've received 2 doses of inactivated vaccine, which was the primary series, and we had thousands of those people in our study. And as I mentioned a minute ago, the Dominican Republic government had purchased the mRNA for boosting in 2021. So we collected blood specimens before the mRNA dose and then periodically after boosting. And we found that 2 inactivated vaccines followed by 1 mRNA booster were just as immunogenic as 2 mRNA vaccines as judged by patients at Yale University. And using the phone, where did that word come from? What word did I intend to say? Using, I don't know what word I thought of using, but let's just say using reactivity to spike protein and receptor binding domain by ELISA as our test done in Akiko Iwasaki's lab by Carolina Lucas. So this is vital information for all nations using inactivated vaccines. And I remember having a very interesting discussion with Akiko, and she said, "Oh, this is very interesting. I think we should submit this to, I forget which journal, Journal of Virology." And I said, "No, Akiko, this is hugely important for those of us who are in the field around decision making, and hugely important for policymakers such as the WHO and Ministries of Health. You should send this to Nature Medicine, which is a perfect journal for that interface between medicine, basic science." She said, "Well, okay, but I don't think it'll be accepted." Well, I was right. She was wrong. We got it into Nature Medicine. And the neutralization titers against Omicron were very low in the persons boosted after coronavirus prime and then boosted with coronavirus, didn't do a thing. Whereas you did get some benefit against ancestral and Delta. But take a look at what happened when you boosted with mRNA. You had a very substantial improvement in response to ancestral, Delta, and even Omicron here by the plaque reduction neutralization test. And if you don't know what that is, I define it here. So this was, we thought, hugely beneficial, and the reviewers of the journal agreed. This has been cited hundreds of times already because I think people found it to be very helpful. And this is a pretty good example of where existing data from Yale were able to be combined with brand new data from Dominican Republic to make possible a study that neither country could have done by itself. We had a second COVID-19 vaccine study, also sort of taking translational science backwards. And that was something that Akiko suggested: do homologous versus heterologous booster doses in those who are fully vaccinated with CoronaVac induce distinct humoral responses. Do responses vary across age groups? And the results were that the IgG against SARS-CoV-2 spike or RBD and neutralization titers against Omicron sublineages were substantially reduced in participants receiving the CoronaVac versus heterologous mRNA booster, especially in recipients greater than 50 years of age. So this was a second study confirming the wisdom of heterologous booster, but also warning that you were not going to get the kind of reaction in older folks. So this was the design that I've already explained to you. Sorry, I didn't mean to do that. Let's go back. This was the design. We also had the AstraZeneca live vector vaccine because that had been tested by Kiko's colleagues. And if you look at the results, you ended up with the Coronavac having an anti-spike and having an anti-RBD effect, but nowhere near as good as either the mRNA prime or the adenovirus vector prime. And this was really the impressive data, which is take a look at how poorly Coronavac boosting did in older individuals. And for those people, not getting the heterologous was putting them in huge risk for adverse outcomes from the circulating Omicron. And I don't have time to go through all of these data, but just homologous regime induced lower virus specific antibody responses with a remarkable impact in lower older participants. There was a lower antibody response that was vaccine specific, not aging per se. And homologous Coronavac booster did not improve neutralization response at all against Omicron sublineages in older adults. And we even had a third study that was conceptualized by Akiko. And she simply wanted the DR data to join with data from all over the world to answer a question: do mRNA vaccines cause fetal defects of anti-syncytin 1 antibody production, which had been postulated by smaller studies? And we found no evidence of fetal defects or this particular antibody induction following vaccination from a wide swath of data from many, many studies. So that was sort of the definitive response to the misleading earlier studies. So that's a little bit about that example of viral translational research. And while you could argue, well, is it implementation science? I think it is implementation science. You've implemented something, billions of doses around the world, and you're asking whether it worked or not. Now let's just do a little side. Let me see my time. Okay, I'm doing okay, but I will go a little quicker now. So I just wanted to share with you a new network that I've become deeply involved with. It's called the Global Virus Network as a vehicle. There are over 80 centers around the world. We have advisors. We have over 40 countries on 6 continents. These are some of the sites. Now, I want to point out that we are weak in the Middle East and the Caucasus region and Central Asia. So I'm actually actively recruiting high-quality virology labs that might be interested in collaborating with our global network from the Georgia region. Now to combat, and I wished I could have been there with you on this visit because that was going to be my side agenda. So I will very much look forward to coming on another occasion. So our mission is to combat viral diseases through international collaborative research, surveillance, healthcare and public health solutions, professional training, public information sharing, and policy guidance. And we're very keen on collaborating. We're not in it for our own glory. We're in it as a network to try to help all the members of the network make disproportionately influential impact. And we've herpes viruses, respiratory viruses, papillomavirus, hemorrhagic fever viruses, hepatitis viruses, including E, rabies virus, enterovirus, and adenovirus, and the different participants. This is not a comprehensive list of all 80. It's just to give you a sense of how we've tried to identify who does what in our network. We have an academy, which is our nickname for the educational enterprise. We've named it after 2 generous donors. And we have a short course, which is starting next week. I'll be in Baltimore on Monday to launch the short course in translational virology. And we have 15 trainees from all over the world who are going to be doing the short course, two intensive weeks. And we welcome you to take a look at our website and see if you're interested in future years. We have a postdoctoral fellowship training program. a Rising Star mentorship program for junior investigators, and David Martinez at Yale was one of our Rising Stars last year. And he's now at Yale. We have an alumni networking series. We even have high school students and college students coming to us. And then we are working on an online short course that we hope to launch next year. And these are just some of our trainees. We have action committees that are trying to facilitate science and application of science in these I won't read them all to you, but in these various topic areas. And these action committees are meeting on a monthly basis, and we have extraordinary speakers, and would welcome you to consider that. We also work with the business community, and we do that quite deliberately because I learned when I was in government, 35 years ago, that neither academics nor government bring products to market. There is no US government or Chinese government or any other kind of government pharmaceutical company or vaccine development company. They can invent vaccines at the NIH or the China CDC. They can develop drugs and immunotherapeutics and diagnostics, but they cannot bring it to market. Only the pharmaceutical industry and the biotech industry are able to bring products to market. So we want to work with the business community. We want to create a safe space for scientists in and out of government to be able to learn from and share with the pharma and biotech industries. And these are a few of the companies that have been working with us, and here are a few of the projects that we've been working with them on, and I won't go into any detail because we want to leave time for questions. So we're working on new regional representatives and centers of excellence. I would love to talk with folks in Georgia about this. Surveillance program, including wastewater, is being expanded. Our online educational programs are being expanded, and we're starting new initiatives in bioinformatics, bioanalytics, and artificial intelligence for pandemic preparedness. And these are some of the organizations that we're reaching out to, some of the companies, academic institutions that are involved, country-specific organizations, regional organizations like the Africa CDC, and other nonprofits such as Gavi, CEPI, and the others. We're going to be going to the CEPI meeting in the very near future. So this is leading me to the final couple of slides. What is the need for translational research in pandemic preparedness? As you all know, a pandemic is a multi-continent epidemic likely to infect thousands or millions of persons. And if you have influenza as an example, a strain can emerge from reassortment of an animal strain with a human strain. If I'm a farm worker and I have human influenza and then I get infected with H5N1 from one of my cattle or cows, I could have a reassortment phenomenon in my body of these viruses and create a very different virus on a human backbone that's highly infectious, human to human. And that's how you have the viral shift, as we call it, as opposed to the viral drift, which is just the small mutations that happen year to year. But if you have a shift, you now have a virus with pandemic potential. And we've had this in the past 100+ years, the so-called Asiatic, so-called Spanish, so-called Asian, so-called Hong Kong flu, and all the deaths, especially from the Spanish flu. By the way, the reason it's called the Spanish flu is it occurred during World War I, and there was severe restriction of what the newspapers were allowed to say, censoring of newspapers in the combatant zones. So they weren't allowed to say that there was this new mysterious, disease that was circulating, killing people. But Spain was not in World War I. So the first journalistic reports of the influenza, the pandemic influenza, came out in Spain. So they got the nickname Spanish flu, which is kind of unfair to the Spaniards. Now, keep in mind that today, with modern travel, 95% of humanity can visit each other within about 48 hours. Sure, you can't get to remote mountain regions or remote jungle regions, but everywhere else you can get to within 48 hours. So it's very easy for, in the incubation period of a virus, for us to visit one place or another. So pandemics are likely to be a lot easier now than they were in the clipper ship days. Then we have densely populated urban areas, and the population exceeds 1 billion people. We had some crowding phenomenon during World War I with the flu pandemic at that time. And we did not have open sharing of information, which inhibited, I think, response. But keep in mind, the population of the Earth was only 1.8 billion back then. So we're a much more crowded planet. So pandemic influenza is a continuing concern. And just looking at this second bullet, with surveillance and recognition, vaccine and treatment discovery and production capabilities, public health infrastructures, global cooperation, political will, funding, respiratory disease mitigation with masks, hygiene, ventilators in hospitals, and workforce deployment. These are many of the things that we didn't do particularly well during COVID-19, with perhaps the exception of vaccine discovery and production. Other things we didn't do so well. And you know that WHO correctly predicts that we're going to have periodic influenza and other pandemics, but there's a lot of uncertainty as to where or when or what. So last slide, collaborative allies, including all of us participating in GIFT, can address a host of global surveillance, control, and prevention activities to respond to pandemic and epidemic threats. We can work with international partners, and the GVN is one partner in translational implementation science research, and I thank you for your attention.